Greetings. Now, this is the Lab 1 lecture for the lab itself. Some of the terms and some of the activities you're going to view or do will be in your lab exercise book. That's the THORACIC book. Some of the activities will depend also on you accessing the lab videos. Many of these exercises have been previously done. Therefore, it's important that you view the video so that you can clearly understand what are the concepts being articulated or demonstrated, and so you can successfully fill out the laboratory handout sheets. They're also called worksheets. Well, let's get started. The objectives here are to review lab safety discussion and complete lab safety sheets. There are no sheets in this case, but I will have you view a couple of lab safety videos just to be aware of the concepts of lab safety, particularly in laboratories such as anatomy and physiology. You are to review Chapter 1, the anatomical terms, and complete the review sheet at the end of the chapter. That sheet has already been posted as an assignment. So you can basically take the sheet, download it. It is in a PDF file version. All of these sheets are going to be in PDF forms. And then you have to just type in and fill out the material. You'll need either Adobe Acrobat, or you can try to fill this out using Adobe Reader, but no matter what form of software that you use, you have to save the document in PDF. If you use Pages, if you use some other open source software, it will not save to a PDF and you will get a 0. I will not ask you to keep repeating the work. You have to know what tools you need right now. You will also review Chapter 4 on membrane transport and complete the review sheet at the end of the chapter. Again, this is in the assignment folder that you will find just below the video for this lab exercise. You will not have to turn it in at the end of class. Rather, you're going to upload this once it's been completed, and all uploads have to be delivered by 5 p.m. on Friday of the week of that activity. I do not take late lab work. Well, let's move forward. Your success in labs will depend on completion of the syllabus exam prior to the lab class. I've already posted that. You will need to complete the review of the lab manual chapters for the lab, as well as review all of the lab videos for the lab class. You need to come prepared for the lab. That will require that you've read the exercises in THORACIC, the lab exercise book, exercise 1 and exercise 4. You should, before you ever enter into a lab, view the videos. Those are going to be extremely helpful. And in every case that you're doing these exercises, just as if you were in a class, in a lab class, you need to fill out the rest of the content in the exercises. If they have questions, you answer them, etc. Don't just focus on filling out the worksheets, because you're going to need these notes to help review before your first lab practical. Okay? We don't have to worry about cell phones upon entry into the class here. Because we are not in an actual lab situation right now, what I'm telling you to do is to review the lab videos that are in the lab video portion for lab 1 or week 1. It's important, once you get into a lab, to know the exit and entryways, to know where equipment is, particularly eye wash station, emergency shower, fire blanket, fire extinguisher, okay, as well as a first aid kit, and also to be very cautious with any reagents and any materials that you use. Never take anything for granted. Do not do any unauthorized experiments. And what I mean by that is, you see some chemicals, don't try mixing them together just to see what would happen. That is an unsafe and in some cases dangerous procedure to occur in a lab. Okay? There is no safety document because we're not having you in the lab. If there was, you would sign it and hand it back to your instructor. But in this case, I just want you to see the videos to get an idea about what is to be expected. Let's get into exercise 1, which talks about anatomical basics. And this is a situation you really have to memorize some of this stuff yourself. But you need to be aware of the anatomical position, hands at sides, palms forward, basically feet slightly astride or basically separated, not at a full attention position where you're heel against heel. When you talk about someone being supine, they're lying down or face up. When they're prone, they're lying down face down. And you can see how these occur, but also the right lateral position, the left lateral position. Sometimes we have to turn someone on their side if they are choking, and they will basically be able to cough up, if they can, that particular item that's causing the blockage. Here we have the diagram that you see, which talks about the anatomical positions. If you talk about things like distal versus proximal, lateral versus medial, think about medial to think about the midline. So if something is moving towards the midline, it's called medial. If something is moving away from the midline, it's lateral. If something is superficial, it's basically at the surface level. Something deep is something that is well below the skin level. And we can be talking about this from the perspective of wounds or location of bones, muscles, blood vessels, etc. It is very important that you get familiarized with these terms right now, as well as craniocephalic and caudal. Posterior or dorsal are basically the same, and anterior or ventral are the same. Now, of course, you got to think about this. Four-legged animals are not the same as two-legged animals. In other words, when I talk about caudal versus cranial, that's one thing, but when I look at my two dogs, they're four-legged creatures. So the medial and lateral are the same, but when you start talking about some of the aspects, because they put their bodies down and are held up by forepaws, it's a slightly different situation when you talk about some of this, as well as structural differences when we talk rostral or caudal. But don't worry about that. Focus on some of the terms that you would see. In this case, you're going to be, interestingly enough, looking at some of these terms, and they do mention some of the other ones, as if you were comparing it to a four-legged animal, okay? Palmar, plantar, rostral are ones that are more referring to dogs, but you're going to see how these more apply to humans as well. For example, frontal or coronal plane, as if you were basically oriented parallel to a long axis. That's where the plane is. So sort of like cutting the person in the front, you get a front view, back view, and basically you're looking internally. Sagittal plane, you have to be aware of the fact, as I've mentioned in the lecture, is that the axis is oriented parallel to the long axis, but it doesn't have to be perfectly midline. If it was perfect on the midline, then we refer to it as mid-sagittal. If it's off of that, then it may be parasagittal, etc. Okay? And then we have, of course, transverse. This is like being sawed in half, perpendicular to the long axis. You know, you've probably seen those magician routines where they have someone go into the box, saw through the center of the box. You can still see them wiggling their toes and they're smiling, but they're separated by space. Well, that's all optic illusion and everything else like that. But I want you to get familiarized with these planes and sections because they're very important when we come to using the terminology of anatomy and physiology. When I talk about sectional anatomy, that I'm referring to the planes and sections. A plane is a three-dimensional axis. A section is a slice parallel to the plane. This is often used to visualize internal organization and structure, and is very important in radiological techniques such as the MRI, PET, and CT. As you can see here, we have organism level and the different types of basically organ systems. And you can see them all around here. The organ system level, integumentary, skeletal, muscular, nervous, endocrine, cardiovascular, lymphatic, respiratory, digestive, urinary, and reproductive. Now, when we go through this hierarchy of organization, of course, you can start with atoms, then molecules, cells, then tissues, then we get to organs, and then organ systems. As you can see, the heart is part of the organ level, and it is part of the cardiovascular system, so that would be the organ system. And all of these organ systems contribute to the functional organism. It is important that you have a basic understanding of each one of these organ systems right now, as well as several notable organs that are existing in various structures, okay, or various organ systems. Here we go again. Surface anatomical landmarks and regions. You got to know them. Just got to know them. We're going to be dealing a lot with these. It's a very conceivable that you will get very familiarized with them. You know, it's kind of like the language of science, or the language of medicine, or the language of allied health. When you look at the terms, they're going to have some redundancy when you go below the surface level. Okay? You're going to find that oleocranial, for example, will exist as a descriptor of a particular bone structure. You will find the acromonial, the same thing. You will find repetition in various structures, whether we're talking about different types of muscle, such as the gluteus maximus. Well, that's in the area of the gluteal, the buttocks. You have all of these terms that I expect you to be familiarized with. And my suggestion is either you take the time and use matching cards, or make a Quizlet on the computer, whatever. But you really do need to understand and be able to intelligently use these terminologies. They're going to be essential for your function and success in subsequent courses that you take, whether they're allied health, nursing, or anything else. Okay? As you can see in the upper anterior view here for the trunk, the umbilical, abdominal, mammary, thoracic, you have also on this limb here the antecubital, brachial, axillary. Not to mention, of course, when we get up toward the neck, which is the cervical, and then above that we have oral, mental, facial, cranial, cephalic, buccal, optic, ocular, or orbital, nasal, and then the frontal. Down here, of course, I'm just going to take one area. You can see the lower part here of the pelvic. Of course, in the lower arm, you have the manual, the palmar, the carpal, the antebrachial, the pollex, the digits. You have to be mindful of also the great toe, the hallux, the digits, the pedal, the tarsal, crucial, patellar, femoral, pubic, inguinal, etc. Going to the posterior view in the upper part of the body, cephalic, cervical, oleocranial, dorsal, acromonial, and in the lower part, of course, lumbar, gluteal, popliteal, calcaneal, and plantar. Translated, know them all. Okay? We also look at the surface anatomy of the abdominal pelvic quadrants. You can see the left and right upper quadrant, the left and right lower quadrant. These are all essential to get down now, because also you have to think about what's underneath them. Usually if someone says, gosh, doc, I've got a pain down here, and you would write down pain in right lower quadrant, a lot of people immediately go, oh, well, maybe that's an appendix. And that would be reasonable. You would not say, gosh, it must be appendix if it's in the left upper quadrant. Then we come into the 9 regions, which include the left and right hypochondriac, the left and right lumbar region, the left and right inguinal region, then through the center part, the epigastric, umbilical, and hypogastric, otherwise known as pubic region. As you can see, the various organs that are separated out in the 4 quadrants and in the 9 regions. Be aware, you need to know where these organs are, because I might ask you, okay, fine. What quadrant is the liver primarily located in? Yes, it does reach over into the left side, but also the right upper quadrant. From here, we start dealing with the cavities, where a lot of the organs and structures are located. In the dorsal body cavity, that consists basically of the vertebral cavity, yes, that's where the spinal cord is. The cranial cavity, that's where the brain is. Then if we look at the ventral body, we're going to see the thoracic cavity composed of the superior mediastinum, the pleural cavities, the pericardial cavity within the mediastinum also. What separates out the thoracic cavity from the abdominal cavity is the diaphragm. Once we get into the abdominal cavity, we have the abdominal cavity, or we combine it with the pelvic cavity and call it the abdominal pelvic cavity. Now, body cavities are essential. They protect organs from accidental shock. They permit changes in size and shape of internal organs. We have the dorsal body cavity, which I've mentioned, consisting of cranial and spinal cavity. The ventral body cavity, which has also been referred to as coelom. Okay, this is divided by the diaphragm into the thoracic cavity, abdominal pelvic cavity. You want to also be mindful of the mesentery. These are important structures that help hold some of these organs in the places of the cavities. For example, the abdominal cavity superior portion, you have the diaphragm to top of the pelvic bones, contains digestive organs. The retroperitoneal space, which is behind this cavity area, is posterior to it and contains pancreas, kidneys, ureters, and parts of the digestive tract. The pelvic cavity is the inferior portion of the abdominal pelvic cavity, and it's within the pelvic bones and contains the reproductive organs, rectum, and bladder. When we get to the peritoneal cavity, it's a chamber with the abdominal pelvic cavity. The parietal peritoneum is the membrane that lines the internal body wall, as you can see here in this diagram. The visceral peritoneum covers the organs, so they're all in contact with the organs here. In the right and the left pleural cavities, these contain the right and the left lungs, respectively. The mediastinum consists of 2 parts. When we start talking about membrane coverings, just to help you, the 2 terms you need to know are basically visceral and parietal. Now with the mediastinum, the upper portion is filled with blood vessels, trachea, esophagus, and thymus. So that's in the middle area here. The lower portion contains the pericardial cavity, which is really what we're talking about, the heart, where it's located. We have other body cavities in the body that you need to be aware of. The oral cavity, as you can see here. The nasal cavity, as you can see up here. The orbital cavities, where eyes are occupying. The middle ear cavity, which you can see right up in here. Okay? And then synovial cavities. Synovial cavities are going to be, for example, where you have synovial joints, you have movement, but you have a fibrous capsule with some fluid that may exist in between the 2 layers here. Now, that causes me to bring up serous membranes a little bit more. These line body cavities and cover organs, they consist of a parietal layer and a visceral layer. They're fluid-filled cavities between the layers. So basically, a parietal layer will line either the cavity or the exterior covering here. The visceral one will be the membrane that kind of directly attaches to this organ right here. There's a space in here filled with serous fluid. Now, as you can see here, you can use the comparison of taking a balloon, sticking your fist into it. The balloon part that wraps around the hand would be the visceral layer, and the exterior part of the membrane of that balloon would be considered the parietal layer. Now, we're going to start moving from there into membrane transport. Membrane transport depends on whether it is passive. Sorry, there's a little typo there, but it is passive or active transport. To help you, passive does not require energy. Active does. Passive transport is molecular transport from an area of high concentration to an area of low concentration. An example of that is you put on some perfume. After a few minutes, the concentration, which is very high around the area of the skin where you put the perfume, the molecules will begin to spread out into the room. Areas of the room that don't have any perfume, they will eventually have some of these molecules migrate out there. But no energy is required to transport them. They're basically moving along what we call a concentration gradient. One area of the gradient is high, one area is low. Molecules will move from high to low. No energy is required. Active transport's a little bit different. It's molecular transport from an area of low concentration to an area of high concentration. That is where energy is required. Think of it this way. If you want to move molecules from an area of low to high, you're going to need molecular pumps. And anything that's pumping needs energy. In a lot of cases, that energy is supplied by the molecule ATP. But we'll get more into this later. What are some of the passive transport types? Diffusion, which is a general transport. This is driven by constant motion of molecules, kinetic energy. In other words, all molecules move around, vibrate, bang against each other, depending whether they're solid, liquid, or gas. And this is dependent on the energy that's present for those molecules. They will eventually spread out from an area from the high concentration to the area of lower concentration. Now, osmosis is a specific type, a selective type of transport. It's passive, but it's the transport only of water molecules. So you have water molecules moving from an area of higher concentration to an area of lower concentration. Now, passive transport depends on either the temperature or the permeability of a membrane. If the membrane separates two sides, one area with high concentration, one area with low concentration, the membrane has to be permeable. Allow the passage through that membrane in one way or another of the molecules that make up this concentration. Now, membrane permeability formats. When we talk about membranes, they can be impermeable, semi-permeable, or freely permeable. Impermeable, nothing passes through. For example, steel. If I have one side with a certain number of molecules and the other side with none, molecules can't get through that steel. Semi-permeable, some molecules pass through. It's usually dependent on the molecular size, the particle charge, and other unique properties. What happens with cells is that some molecules can pass right through a cell membrane as long as they are nonpolar, have no charge on them, meaning that they're ionic charge or something like that, and if they're relatively small in the molecular size. Something that's freely permeable, all types of particles will pass through the membrane. That's something that you do see in certain cases. Think about it almost as if you have a sieve where you have the size being much larger than many particles like sand or dirt or something else like that. or water or something else like that will pass through. Okay? Freely permeable does mean, though, that it's unlimited. Anything can pass through, no restrictions. Now, when you review the lab manual membrane experiments, you will see some representation of this in lab videos. Remember that I said it's either the permeability of the membrane or the effective temperature that will allow you to have simple diffusion. Well, you could take a glass of water, and at room temperature the water is presently at, and you can take another glass of water that has been chilled to 35 degrees. You take dye tablets. Oh, come on, everybody knows what dye tablets are. If you remember those little tablets that used to stain the outsides of Easter eggs, for example. Or you could take a food coloring drop. You put drops in. The temperature has a direct effect on the transport, that is the diffusion of molecules. The dye molecules will travel faster when you have a higher temperature. Room temperature, the molecules will move at a certain rate. At a very cold temperature, the molecules will move at a much slower rate. If I could take that glass of water and heat up the water to, let's say, 150 degrees, and I put in the same drop of dye, the molecules will move really a lot faster. Now, unfortunately, we don't have the lab equipment in front of you, but we do have some videos that I hope will help this. But basically, when you talk about diffusion across membranes, you have to have, as I mentioned before, two requirements. A concentration gradient that must be present, and a substance that must be permeable to the substance, membrane that must be permeable to the substance being transported. Here's an important point. When you do the IKI experiment and the starch, etc., let me help you with what IKI is. They call it potassium iodide. It's really called Lugol's iodide. And what it is 1% iodide solution, 2% potassium iodide solution mixed together. What happens is when this solution comes in contact with starch, the solution, which would be somewhat like a purplish color, turns black or a dark blue-black color. Now, starch is a very, very large molecule. Starch will normally not go through dialysis tubing, either from outside to in or inside to out. Potassium iodide will go through. So whichever compartment, either inside of the dialysis tubing or outside of the water where the dialysis tubing is being held in, wherever you have molecules that are small enough, they'll go through the dialysis tubing and come in contact with the material inside or outside of that dialysis tubing. Now, osmosis is diffusion of water through a selectively permeable membrane. A solution that is a mixture that consists of a solvent and a solute. Let me help you there. Solution. Solvent, what constitutes a solvent? Anything that is in greater amount. Solute, anything in lesser amount. That's confusing. Not really. You've probably heard of saline solution, all right? We use that in various medical applications. Well, a saline solution, the solvent is the material that's in greater amount for this mixture. The solute is in much lesser amount. So water is the solvent, and the small amount of salts are the solute, okay? Same thing you can do with a lot of other solutions. Air's a solution. It's a mixture of gases. The solvent, the greater amount, is nitrogen, which is at 78% of the gas that makes up what we breathe. The solute, the lesser amount, would be oxygen, water vapor, carbon dioxide, and a variety of other Nobel gases and other things. Okay? Now, water is considered a universal solvent. That's in part why astronauts and space explorers and they're sending probes everywhere. They're trying to find water, not only because it's essential to live on, but usually it's believed that if they find water, they find a higher probability that there may be the potential for life there. Now, solute concentrations can be measured in milliosmoles. Now, we're going to probably see this as M, a small m, a big O, and a small SOMOL. Excuse me. Osmoles are a measure of the solvent, of the solute. Okay? Osmoles are the measure of the solute. Milli, the prefix, is always saying 1/1000ths. So what you have is a measurement which is the number of dissolved particles per, in this case, milliliter of solution. Okay? So if I have a salt solution, say 10%, that's saying in the milliliter solution, I've got basically 10 milliosmoles, okay, of salt in there. I hope that makes things a little bit clearer. A lot of times we compare one solution with another, especially if they're on different sides of a membrane separation. You just can't sit there and say, well, this is a hypertonic or an isotonic or a hypotonic. Compared to what? You always have to have some other solution you're comparing it to, whether it's blood, whether it's pure water, whether it's cerebral spinal fluid, etc. Okay? So when you use the terms hypertonic, isotonic, and hypotonic, these are terms that compare the solute concentrations of two solutions. If you have a hypertonic solution, that's going to have a higher concentration of solutes than your reference solution. If you have a hypotonic solution, they will have a lower concentration of solute than the reference solution. If you have an iso, now remember, iso usually means same. Hypo means lower. Hyper means higher. Okay? Each one of these terms gives you a concept of the amount of the tonic or the tonicity, the concentration present. Isotonic solutions have the same concentration of solute as the reference solution. What do I mean by reference solution? Okay, well, let's go over here. If you go to the exercise, you're going to see things like the experiment using molasses. Well, molasses is a very concentrated sugar solution. You'd call it hypertonic. Okay? Have you ever seen it? They put this into basically a thistle tube. They take the thistle tube, it's got a membrane wrapped around it, and in the wide part, the bell part of the thistle tube, they fill it with molasses. Now that's very, very concentrated. On the outside, they take the thistle tube and put it into a very large flask of pure water. Why? Well, where's the water concentration the greatest? Surrounding the thistle tube. Where's the water concentration the lowest? In the molasses. Where's the concentration of solutes greatest? In the molasses. Where's the concentration of solutes the least? In the pure water. So water is going to go in the direction from high concentration to low concentration. It's going to go in the direction of going right through that membrane, right into that thistle tube. As it does, you're going to notice that the rest of the stem of the thistle tube begins rising up as water rushes in and tries to move into this very, very hypertonic solution called molasses. Okay? Now, look at some of the videos. They will be repeating these very same experiments. Okay? There's nothing to hand to me about the lab safety form, but I do want you to see the lab videos. Okay? And the other thing that I want you to do that will help is review chapter 1, complete all of the written parts, including the handout, or they call it the worksheet exercise. I've already given that to you by scanning it in, setting it up as a form. Just when you bring up your cursor to that particular area, you can type in the answers, okay? Usually you've got to have either Adobe Acrobat, or you may be able to use it with some of the free softwares out there that will allow you to basically make a free PDF. Sometimes you can also download it into Word, but then save it as a PDF, okay? Same thing with chapter 4. Do both exercise 4.1, 4.2, complete all the work, complete the form. You don't have to hand it in to me. You do have to upload it at the Blackboard site, okay? Look at all the videos and review the lab manual for the next set of exercises, which is going to be exercises 2 and 3. And I hope you all have a nice day.